Hello, welcome to this webinar entitled Development of Implantable Devices for Sustained Drug Delivery. First of all, I'm going to introduce myself. My name is Dr. Eneko Larrañeta, and I am Senior Lecturer in Queen's University Belfast in the School of Pharmacy. Okay, before we start with the topic, I'm going to introduce myself a little bit. I am Spanish, and I am chemist by training. I came from the north of Spain, from a city called Pamplona, and there I did my degree in chemistry and my PhD in chemistry. I work on the development of self-assembled gels for drug delivery purposes. Subsequently, I started a postdoctoral position under the supervision of Professor Juan Manuel Irache, developing oral drug delivery systems, specifically nanoparticle systems. Okay, that gave me a new point of view, because obviously I came from a chemical background, so I start to have expertise and to start thinking on drug delivery systems from a pharmaceutical perspective, okay, not only from the chemical perspective. Subsequently, I moved to Belfast, okay, where actually I still work, and I started a postdoctoral position under the supervision of Professor Ryan Donnelly on the development of polymeric transdermal drug delivery systems, more specifically microneedle drug delivery systems, microneedle systems for drug delivery. It was in 2016. I worked there for several years until I got my independent position in 2016, where I started my own group, and I started to work on the development of implantable medical devices for drug delivery purposes. Okay, and currently I'm working in Queen's University Belfast as a senior lecturer, okay, in the School of Pharmacy. The picture that is on the website or in the previous slides a little bit old is when I first started. That's a more recent picture of me, just in case you see me in conferences or in out there. Hopefully, as soon as we are allowed to go out to conferences and you want to discuss an idea or grab a cup of coffee, or even better, a beer after the conference to discuss any idea or any potential projects or collaboration, there you have a picture, and you can see how I look. You can just approach me at any time. So after this brief presentation, I'm going to start describing and start talking about the topic of today's webinar. Okay, that is development of implantable devices for sustained drug delivery. I keep it broad because in my group we are working on the development of different types of implantable devices, not only the conventional subcutaneous implants that maybe everybody is familiar with. We are working on some implants for cardiovascular applications. So in this case, I wanted to keep it a little bit broad to provide information about all these systems. Okay. First of all, why are we interested in my research group on the development of implantable drug delivery systems? Basically for two main reasons. First of all, we have the main reason that this patient, okay, there is massive problem with patients' adherence to treatment, okay, especially whenever we have patients suffering from chronic conditions. They require constant uptake of medication. And in a lot of cases, the patient is not following the rules because the patient forgets to take the medication, or because the patient don't want to take the medication or the treatment, and that has a clear impact on the quality of life of the patient. And actually, it can affect massively not only the quality of life. In some cases, it can even lead to the premature death of the patient. And additionally, it has an impact in the healthcare systems, contributing to an added cost, okay, of the treatment of the non-adherent patients. And additionally, obviously, there is a clear market value. Okay, there is a clear industrial interest on the development of new type of drug delivery systems. So these two pillars makes this topic quite interesting for us. As mentioned before, one of the key pillars is the patient. Why we want to develop implantable drug delivery systems? To provide unattended drug delivery to patients, and this is especially important in chronic patients suffering from from chronic conditions, okay? Because adherence to treatment is a massive problem. In the graph, you can see some stats and some information about the cost of non-adherence to treatment from all the conditions in general globally as a problem, and for example in America and in UK specifically. For example, in the UK, the cost of non-adherence to treatment is up to £500 million. Okay, if we focus on specific conditions, just to give you a little bit of an overview, for example, some studies said that 50% of patients discontinue treatment or like not adhere to treatment. But for example, for conditions like schizophrenia or Parkinson's, there is studies where they suggest that up to 70% of patients, or around 70% of patients, that is a high number, okay, discontinue treatment or they not adhering to treatment properly for schizophrenia and Parkinson's disease. And it's important to remember that in conditions such as schizophrenia, if a patient is not adhering to treatment, this can put the patient at a high risk of relapse and hospitalization. It can increase the risk of suicide, and it's estimated that each patient that the cost of a relapse to the NHS is around £15,000 per year and per patient. Okay, that's the cost of a relapse. So if we can get implantable systems or like long-acting systems that deliver antipsychotic drugs to schizophrenic patients, we can just save that cost to the healthcare systems. In the case of Parkinson's disease, it's estimated that the hospitalization cost in England specifically are estimated to be 194 million per year for Parkinson's disease patients. Okay, so obviously this is there is like a clear problem that can be solved with the technologies and the devices that we are going to describe here. And obviously there is a potential to save resources for the healthcare systems, and is a potential to improve the quality of life of patients. In addition to the patient needs, there is a clear growing interest on this type of systems, as it can be seen in the graphs that I present in here. If we cross the terms drug delivery and implant in a scientific portal, like for example Scopus, you can get or Web of Knowledge, you can get this type of graphs, and you can see that there is a growing interest because the number of items published per year is increasing since 1990. Okay, increasing every year. Okay, and the same if we cross the terms drug delivery with long acting. Okay, so we can see the same trend. And additionally, in addition to the academic interest, there is a clear interest from companies, because there is like several products and new products are being added or like studied in clinical trials every year. And we have at the global control release market value that is 37.8 billion of United States dollars in 2019, is expected to grow at a rate of 7.8% up to 2027. Okay, so there is a clear interest of the market in this type of devices, in implantable devices and long-acting drug delivery systems. So if we combine the need of these type of systems from a patient point of view, and we combine this with the academic and industrial interest on this type of systems, that's what makes this topic highly interesting to us. Okay, and that's why we are working on the development of this type of drug delivery system. Actually, as I mentioned before, there are several devices, implantable devices out there in the market, a good few of them. Okay, and there are different shapes and sizes for them. Okay, probably one of the most well-known implantable devices is like Nexplanon. Okay, it's like the contraceptive implant. Here we can see the applicator. That's just actually an example of the implant and how it's applied. Okay, it's subcutaneously in the arm and can provide long-acting delivery of contraception for several years. Okay, but there are other commercial systems, like for example the wafers from Gliadel that they're implanted in the brain for the treatment of glioblastoma, okay, brain cancer. They can provide the delivery of a chemotherapeutic agent locally, okay, for a sustained period of time. There is other devices like stents that they are implantable devices, okay. Normally they are used for expanding occluded arteries, and drugs can be deposited in the surface to provide a more sustained sustained drug delivery locally. Okay, there's other examples of implants, subcutaneous implants, or just implants like here we have like an example of a reservoir type implant that you can find in the literature. Okay, so there is interest academically and industrial, and there is a wide variety of commercial products that are based on implantable drug delivery systems. So obviously, considering all these factors, that's why we are interested in the development of this type of technologies. Obviously, here you have a quick summary about polymers, conventional polymers that they are used to develop this type of implants, a wide variety of manufacturing techniques, and again, we can these type of devices from a broad perspective can be applied in different locations in the body. It can be intracranial implants, like we saw for the treatment of cancer treatment. It can be intravaginal implants, subcutaneous implants. So they are quite common, as I mentioned, especially for contraception, or intraocular implants. But there are other ones that are not included here, like cardiovascular implants and so on. Okay, so in focusing on this topic, I'm going to start describing some of the work that we have been doing, and among the manufacturing techniques, I'm going to be focusing on the use of 3D printing. Okay, 3D printing is a great technique that can be used for quick prototyping of medical devices and drug delivery systems. Okay, so it allows researchers, especially in labs, like to prepare customized devices in a quick way. But otherwise, it will take time and it will take a lot of resources. For example, if we need to just use injection molding, we will need a new mold every time we want to change the design of a medical device. So it's a really good technique. It's a great technique, and it can be easily scaled up. But 3D printing is allowing us to design different type of prototypes and optimize the geometry and other parameters like drug loading and so on in a really quick way. Okay, to do some research. 3D printing is a method of manufacturing that is also called additive manufacturing. Okay, and why it's called additive manufacturing? Because basically it's used to add layers of material in a sequential manner to form a 3D object. Okay, as opposed to subtractive manufacturing. Okay, subtractive manufacturing is instead of adding layers of material, the layers of materials are removed from a block to obtain a 3D object. Okay, this is quite a traditional way of manufacturing. Okay, and if you want to go, you can see in the Greek sculptures, or like in the Renaissance amazing sculpture, like for example David from Michelangelo. That's like a way of doing subtractive manufacturing. Okay, we are going to focus in additive manufacturing for the following examples that I'm going to describe. So, one type of additive manufacturing or 3D printing is FDM, that is fused deposition modeling. Fused deposition modeling works by pushing a filament of a thermoplastic to a hot end, a heated nozzle, and the nozzle is going to be adding the layers of molten material that it will solidify after cooling down. Okay, here you have a quick clip video of how it works. Okay, this material, we have flexibility on the type of material. We need a thermoplastic. Okay, and actually different substances can be added here. You can add different type of drugs or different type of molecules to change the properties of the thermoplastic, or in some cases, if we are working in drug delivery devices, just to incorporate a drug into a 3D object. In this work, in the first example of work that I'm going to describe, is one of the first works that we started when I first started working on implantable drug delivery devices. So we decided that a good way of experimenting with the technique will be to prepare some reservoir type implantable devices using 3D printing. So basically we will print the case of an implant, and we will fill some model molecules inside to study the release of these molecules. So we have two different main types of implantable devices for drug delivery. We have the matrix type implant, where the drug is just dispersed on a polymeric matrix, well, polymeric or normally it's a polymeric matrix. Or we have a reservoir type implant, in which, you know, a case or like a container, it's made and the drug is located inside. So in order for the drug to diffuse out, the membrane should be semi-permeable, or some orifice or opening in the casing or in the implant. The implant cover should be present. Okay, so what we did is design different type of implants, as you can see here, and we use two different type of materials: polyvinyl alcohol and polylactic acid. They are both biocompatible, and actually polyvinyl alcohol is soluble in water, so it will be soluble inside the human body, and polylactic acid, it's biodegradable. Okay, maybe you are thinking how you manage to do implants like this with this geometry and two different materials. Basically because we use a dual extruder FDM. So that's a 3D printing device, 3D printing machine, that instead of only adding one type of material is capable of being used to add two different type of materials, as you can see in this image. Okay, by using two extruders, two hot nozzles. Okay, so for this purpose we use, as I mentioned before, PLA and PBA. PLA is polylactic acid, okay, and PBA is polyvinyl alcohol. PLA, as I mentioned, PBA is a soluble polymer, while PLA, polylactic acid, is biodegradable. Okay, just is a simple degradation process. Water hydrolyzes the ester bonds, releasing, you know, lactic acid monomers, and just the polymer is degraded slowly. Okay, this polymer is quite well used not only for medical applications or pharmaceutical applications, but is used in other things. For example, you can see an example of a bottle made of PLA. You can see how it biodegrades, just, you know, over time. So after preparing this type of implants, what we can see is like what we were capable of printing this type of implantable devices. Here you can see a picture of one of these implants in the hand of one of my group members, and actually we did a little bit of imaging, because these implants have certain windows in their design that they were made of PBA. Okay, these windows will dissolve slowly, or not that slowly, depending on the time, and will allow drug release. Okay, the implants will be filled with different compounds. In this case, for this work we use model molecules, use highly soluble molecules, because one of the challenges that we wanted to tackle is sustain the delivery of highly soluble drug molecules. That's challenging. Okay, hydrophobic molecules, due to the solubility, the release of these molecules is normally by nature slow, by the nature of the compound, but is challenging to sustain the delivery of hydrophilic compounds. Okay, so basically we use methylene blue because it's easy to analyze and it's a really good model and it's highly soluble, and we use ibuprofen sodium and ibuprofen the free base. Okay, the free base is not that soluble, but we wanted to do a quick comparison. So what we saw is, like, we can see the different windows that we have. In this case we have a 1 by 1. Technically it should be a square, but during the 3D printing process we have like some distortion. We have like the 1 by 1 window of PBA, and here we have a large window of PBA. Okay, and the micro-CT analysis can show that the different cross sections of the implant filled with these molecules. Additionally, we studied the dissolution of these PBA windows as a function of the time, and we use even optical coherence tomography to evaluate the dissolution in water, in just in PBS, not water, in aqueous environment. The dissolution of these windows was quite fast. Okay, in 35 minutes we can see that the windows dissolved completely. Obviously, in biological tissue, the availability of fluid and the diffusion of the polyvinyl alcohol will be slower, but we anticipate that it will happen quite fast, and the drug will be allowed to permeate and to be released from this aperture. So whenever we studied the release, we tried different implants. tried implants made of PBA, so they are highly soluble and they will dissolve and release the drug immediately. Here we have methylene blue release. In 24 hours you can see 100% release from soluble implants. That's expected. We have a large soluble window from the implants. You have a slower release, but still quite fast. In 4 days, all the drug cargo will be released. So this makes these implants not really useful for sustained drug delivery. Subsequently, we have implants with little windows, soluble windows, but spaced evenly. That implant will provide a more sustained drug delivery, but still, we will get only up to 1 week of drug release. Whenever we start to reduce the number of these windows, we can see that these implants are capable of providing a more sustained drug delivery. Okay, and actually there is a clear correlation on the drug release in terms of milligrams per day that they are released, and there is a correlation of the milligrams per day release with the window size. Okay, the release window. This was really promising, considering that methylene blue is a highly soluble compound. We can get like up to almost a month of release, 25 days. Still, for certain applications will not be great. Okay, but at least by tweaking the design of the implant, we can affect the drug release rate. We believe that the testing method that gave us like 25 days of release is not the most representative, because the implants were placed in PBS, phosphate buffer saline, and that's not a really representative method. That's not a representative conditions of the in vivo situation. So what we decided is just to study the release on an agarose gel, because we saw in the literature that this method was tested before to study the release of certain compounds for drug-loaded implants. So we pick the implant with one by one window, a soluble window, and we load methylene blue. Okay, so you can see that the release is more sustained. Well, after 3 days, well, it don't look that. Maybe in these pictures of the agar plates don't look really sustained. After 3 days, we have the plate completely blue. But if we quantify at different times, at different distances, different times, we can see that the system continue releasing for 42 days. Okay, for 42 days, you can see that obviously the methylene blue diffused through the tissue, and but it continue being released for 42 days. So that is a more respectable time frame. Obviously, this type of implants cannot be used for for therapies that require long-term therapies, but they can be used for for example, for applications such as loading antibiotics to prevent infection after surgery, or for example, they can be used for the release of for example, anesthetic like lidocaine to prevent pain after major surgery, because these implants can be put in place and they will be biodegradable, so they don't need to be removed after being put in place. Okay, but we were not satisfied with these time frames, so we decided probably the best way to sustain the release of hydrophilic molecules will be to use coatings, okay, or specific membranes that will limit the permeation of the drug. Okay, I'm showing here only the results of methylene blue, because they were the best model in terms of study this permeation. Ibuprofen, due to a high ibuprofen, especially ibuprofen sodium, the salt was heavily soluble, and the release profile were really fast. So obviously there's a challenge there, and this type of implants were not suitable for that type of compounds with that high solubility. For this purpose, we continue using these implants with a large soluble window. We focus in that design. What we do is coat them with specific formulations. In this case, again, we are interested in having biodegradable implants. So we coat with some formulation based on polycaprolactone that we designed in this lab. Actually, all this work, the previous work and this work was developed by my PhD student, my former PhD student, Sarah Stewart, now Dr. Stewart, during, as I said, during her PhD. Okay, she did like a really, really good job of putting all this together. So she did like a formulation work to prepare different type of membranes that are going to sustain the delivery of hydrophilic compounds from this type of implants. So for this purpose, as I mentioned, we use the implants that were used were like the implants with a large window, soluble window, and basically what they were filled with model compounds, with the same model compounds that we tested, that I described in the first example, and they were coated with a formulation based on polycaprolactone. But different type of polycaprolactones were used. We use high molecular weight polycaprolactone, and we combine it with a low molecular weight polycaprolactone, and in some formulations we included polyethylene glycol. Okay, the different combinations of these polymers they gave different properties to the membranes in terms of porosity and drug permeability. Okay, so what we saw is like obviously the porosity of the implants increases as a function of the time. Okay, here you can see an example of a porous membrane at the beginning of the study, whenever it was freshly prepared, and after like a few months, you can see that the porosity increased because the biodegradable nature of the materials. In this case, you can see a microscopic image of this type of systems, in which, like, you know, we have hydrophilic pores and hydrophobic matrix. Okay, this was used 2 different fluorescent dyes to highlight that, and basically the porous nature of these membranes will make them ideal for drug delivery purposes. It's interesting to know, for example, that the inclusion of hydrophilic fillers like polyethylene glycol in the membranes will give higher porosity, because obviously these fillers will be dissolved. Filler or like of this compound, polyethylene glycol, will be dissolved after a while, after 60 days in water or in aqueous environment, and will give a higher porosity. That this is not observed. This increase in porosity was not observed for the membranes that did not contain polyethylene glycol. The implants were coated with these formulations and characterized. Here you can see like some cross section of the implants with the coated implants, some pictures of these implants, and some OCT image highlighting the different type of sections. The coating in white, the PLA casing of the implant, or the PLA implant in blue, PBA window in red, and just the drug core in different colors. In here is blue and green. Okay, so micro-CT was used to characterize them in terms of measure the thickness of the coating and the dimensions of the implant, and the methylene blue release was evaluated in these implants. Interestingly, the films that were prepared using high molecular weight polycaprolactone, they didn't provide any release. They were quite water impermeable, but as soon as low molecular weight polycaprolactone was included in the formulation of the films, the release rates were different, were increased, and actually we can see that by changing the proportion of polyethylene glycol, low molecular weight polycaprolactone, you can have different types of release. Additionally, you can see that adding polyethylene glycol provided a faster release, but there was not a massive difference when comparing with films made of 40% high molecular weight and 60% low molecular weight polycaprolactone. The most interesting film was the one that was prepared using 50% high molecular weight polycaprolactone and 50% low molecular weight polycaprolactone, because it provided a sustained delivery of methylene blue for up to 300 days. I think the graphs continued in the last version that I don't have the last version, but if you're interested in having a look at this, it was published in Molecular Pharmaceutics. You can find this in that journal, and you can see we reached to around 300 days of release. Not everything were good news. For example, ibuprofen sodium, again, due to the high solubility of these compounds, we couldn't get like sustained release profiles in this case, because the water permeation inside the implant, the water that can permeate inside the implant was enough to dissolve the majority of ibuprofen, and the release was quite fast. So this means that these membranes are promising, but obviously they need to evaluate it for specific examples. For each drug that each drug that can be loaded in implants, the release needs to be evaluated, and the release rates need to be checked carefully, because obviously the solubility of the drug will influence massively on the release rates. This work was included in 2 publications, one in Pharmaceutics that describes the 3D printing and characterization of the implants with different soluble windows for drug release, and a second one in the journal Molecular Pharmaceutics describing the development of the coatings for preparing the rate controlling membranes for prolonged drug delivery of hydrophilic molecules. And if you want more details about these works, that you can find them in these 2 articles that actually both are open access, so it should be easy to access. In addition to the use of FDM for the development of implantable devices, there is other type of work that we have developed in the group. In this case, we use pastes, high concentrated dispersions or solutions of polymers and drugs to print with using extrusion-based 3D printer, but in this case, this printing process don't require high temperature. So in this case, we tried 2 projects. One to create implants, reservoir type implants. So in this case, we will have a membrane. So the membranes that we developed previously, we use them again, but before using the membrane, we printed like an implant containing an antipsychotic drug, olanzapine, and using high molecular weight polyethylene glycol. Well, technically it's called polyethylene oxide, because in this case the molecular weight was 100,000. And in this case we have an example of the implant that was printed, and here you have the implant coated in the film. In this case the implants were not coated. Basically they were wrapped in the membrane and sealed thermically using some hot clamps. They were sealed inside. Here you can see like an image of the optical coherence tomography of the implant, and the implant coated. Here you can see the coating. And additionally, we use similar approach, but instead of using an aqueous suspension of the drug that is hydrophobic and polyethylene glycol, we use an organic solvent, dichloromethane, to dissolve polycaprolactone, and the drug, and printed some matrix type implants, as you can see here. So this type of implants, they are not coated, they are matrix type or monolithic implants. The drug is dispersed within the polymer. Obviously, it's not as environmental friendly or as patient friendly because of the use of solvent, but technically, if we make sure that the solvent is well evaporated, and that is not should not be a problem for the application of this technology. As DCM is used, dichloromethane is used for certain specific pharmaceutical applications. And if the levels of the residual levels are controlled, there is no problem. So basically what we can see in the reservoir type implants, we have a sustained delivery of olanzapine for around 200 days. We have 2 drug loadings. You can see that the drug loading influence in the reservoir type implants the release. Higher drug loadings will be translated in a more concentrated or saturated environment inside the membrane, inside the pouch of the polymeric implant, and it will end in a 0 order release, while lower drug loadings, as you know, the supersaturation or the saturation inside the implant will not be maintained through all the process. We'll have like different type of release profile. Additionally, in the matrix implants we have a sustained delivery, but in this case the formulation was slightly different. We have the 50%, the blue plot represents like full polycaprolactone olanzapine mixtures containing 50% of drug, and the red one that shows a higher drug release. For that purpose, in order to achieve a higher drug release, polyethylene glycol low molecular weight was included in the formulation, so obviously it's going to generate a more porous structure during the procedure because of the hydrophilic nature of PEG, and it's going to be translated in a higher drug release. In order to get a more sustained drug release, by increasing the percentage of drug and in the implants containing polyethylene glycol and polycaprolactone, we get like a linear release profile. So that shows that this technology can be used to tweak the release from the implants in a really easy way, because in a really simple way by using different paste formulations. Obviously the devices need to be dry before they can be used. In addition to those type of implants, we developed finally another applications of the films, of these rate controlling films, and FDM 3D printing. In this case, we create a new type of films that they were made instead of being made of polycaprolactone. That polycaprolactone has a low melting point of around 55 degrees Celsius. As soon as you try to print around those membranes with materials such as PLA, that has a melting point of around 180 or around that temperatures, it will be melted. So what we decided is to create some PLA-based membranes, and in order to be useful for drug delivery, like as you can see here, have highly porous membranes by combining PLA with low molecular weight polycaprolactone. So in these type of membranes, we can print on top of them. So we use FDM, we fixate these membranes on the building plate, and we print on top of them to create implantable devices with the film fixed and attached to one side of the implant, so the implant can be filled with drugs. In this case is tetracycline, formulation containing an antibiotic, tetracycline, and sealed in the other side. Okay, so this type of implants can sustain the delivery of this antibiotic. This can be helpful, for example, to prevent infection after surgery, and by modifying the drug loading, the amount of drug inside the implant, the release profiles can be altered or can be modified. Okay, we have quite sustained delivery whenever the drug delivery 60 milligrams or but this tells us that the membrane is capable of sustaining the delivery of hydrophilic molecules again, and we can use 3D printing to quickly prepare some prototypes on top of those membranes. So far we have described the use of 3D printing and different formulations and approaches to create different type of implantable devices for sustained drug delivery, but we have not mentioned any specific conditions. Okay, we have, before we mentioned the use of olanzapine as a model molecule for testing the implants, that is an antipsychotic for treating schizophrenia, but we have not focused in any particular condition. Now we are going to talk about implantable devices for the treatment of hypothyroidism. Okay, hypothyroidism is a condition that is globally affecting around 200 million people. This is affecting is more common among women than men, and it has different type of symptoms that they are not life-threatening, but if the condition is not managed properly in the long term, it can be serious complications. Okay, like in the death of the patient, or like mixed epidemic coma. Okay, so it's really important that this condition is treated, and one of the limitations of the current treatments of the condition is that it requires levothyroxine. Okay, levothyroxine is quite potent. We require 100 micrograms, okay, per tablet per day, and actually that makes this drug an ideal candidate to prepare an implant for sustained drug delivery. Not only that, is because hypothyroidism treatment, the therapy using levothyroxine is challenging, because like the absorption of the drug can be affected by food, by the food intake. It's not ideal just for the patient, because it needs to have like a certain routine before taking the tablet in order to ensure that the absorption of the drug is going to be consistent. So this problem makes levothyroxine an ideal candidate to be included in an implantable device. In this way, the patients can have a continuous and sustained drug delivery for prolonged periods of time without relying on just taking the levothyroxine orally that can have absorption problems. That's why we developed these type of systems. In this case, we don't use 3D printing. We use like a solvent casting approach, and we use polycaprolactone. Why? Because we decided that the materials that we, after developing those films based on polycaprolactone, we decided that the material has really interesting properties, and we wanted to continue exploring it. Okay, so actually in this case we prepare and test the implants in vitro, and subsequently we did an in vivo animal test. So to start, well, I mentioned that we didn't use 3D printing. We used 3D printing to create some molds, some master molds, to create finally some silicone molds that allow us to get our implants. Okay, for this purpose we 3D printed a master mold using PLA, polylactic acid, and later cast silicone on top of that. And what we do is like use solutions containing as I mentioned before, some dichloromethane solutions containing polycaprolactone and the drug, okay, in this case levothyroxine, to cast inside the molds and get the implant, the rods that are designed for subcutaneous implantation. So here you have some pictures of the different type of formulation. We tried to use two different drug loadings, 20% and 40%, and we use formulations that contains 100% of the polymer was high molecular weight polycaprolactone. I think 50 kilodalton polycaprolactone, and later we tried different formulations containing low molecular weight polycaprolactone of around 500 dalton. Okay, so the combinations of because we have interesting results in the past with this combination of polycaprolactones, we wanted to see the effect in drug delivery. So you have different type of implants, and actually here you can see that we incorporated polyethylene glycol. Okay, because we know that the incorporation of this hydrophilic polymer into the matrix can alter the release profile, and in this case you can see that all the implants look like whites or around whitish gray, the color between white and gray. Here you have an implant and besides a euro coin to see a little bit the dimensions of these rods. Basically, what we observed as soon as we prepared implants including polyethylene glycol in the formulation is that the color was completely different. So we anticipated there was like a degradation there. And actually that was the case. We didn't even bother in preparing like a one with higher levothyroxine content, because we can see that polyethylene glycol was like degrading the drug inside the implant. We established that probably levothyroxine is quite susceptible to humidity, and polyethylene glycol is going to contribute, it's going to make the implant attract water content, and ambient humidity, and that's what is going to lead to the degradation of the drug. So whenever we observe the implants on the microscope, you can see the crystals of the drug, because obviously whenever we cast the solutions, levothyroxine is not soluble in DCM, so basically the polymer was dissolved but not the drug, so it was a dispersion with a spit mixer to mix these formulations. And it was clear to us that apart from the pictures, that implants containing levothyroxine presented some sort of problems, because the drug crystals have different shape and the polymer thing was completely different than the other type of implant. So that's put us in the direction that thinking that there was a degradation, and actually there was a significant degradation of the drug. Whenever we analyze using HPLC all the implants, we can establish that we have recoveries of the drug higher than 85% of the initial drug content. Here it was not the case, so we decided not to use this type of implants and not pursue the use of polyethylene glycol in the formulation. Actually, this was the beginning of some sort of challenges or problems. This work was developed by my former PhD student Sarah Stewart, and actually she had like a tough time working with levothyroxine, because if you have ever worked with levothyroxine, you will know that it's a highly unstable compound. The stability of this compound is quite challenging, but she worked hard and she was successful in stabilizing the drug. well, the implants was fine because there was no humidity, but it's quite challenging to test this compound in an aqueous environment for doing in vitro testing when it's a highly unstable compound in aqueous environments. Okay, so we try different type of solvents and co-solvent trying to stabilize them, and finally we discovered that the best molecule was like BSA, or in other publications you can find that human serum albumin works too, so albumins. Why? Because this drug has a high binding to proteins, so it's stabilized. So it was achieved, 14 days of stability were achieved, while if the protein was not present in the aqueous environment, the drug was quickly degraded in a few hours. Okay, so to do implantable devices, you need at least the drug to be stable for at least 24 hours to be able to quantify it in the release medium. So obviously it was not easy, but it was achieved that the stability increase of the drug was achieved by adding bovine serum albumin, and the stability was tested for up to 14 days. Subsequently, we tested drug release profiles of the drug from the different formulations, and it was established that the most appropriate formulation was the one that contains 60% low molecular weight polycaprolactone and 40% high molecular weight, because we have a more sustained delivery over periods of for up to 100 days. Obviously, we picked that formulation. We decided to continue using that formulation and use it in an animal experiment, and we use the formulation containing only 100% high molecular weight polycaprolactone as a control. So we observed that in the formulation that contain a mixture of high molecular weight and low molecular weight polycaprolactone, we have a higher area under curve and a more efficient delivery of the drug, while in the formulation containing only high molecular weight polycaprolactone, we have lower levels of the drug in plasma. Obviously, this test was done with a half an implant. Okay, we cut the implant in two, and by half, and rats were implanted with half an implant. Obviously, this is just like a preclinical study to see if the implants were capable of sustaining the delivery. This test we achieved at least 28 days of release of the drug. More tests needs to be done because obviously the dose was not adjusted accordingly to the rats, because the implant will be tiny in that regards. So the results suggested that the implant that will be needed to provide therapeutic doses will be at least 10 times smaller than the ones that we administer to the rats. So that will lead to a really small implants, and they will open the possibility of using minimally invasive implanting techniques to apply implants of this size. Specifically, the size of the implants, based on these results, will not be 10 times smaller. Sorry, that I mentioned just a couple of minutes ago. Will be between 5 and 14 times smaller than the ones that we applied into the rats. Okay. I mentioned that systems for like minimally invasive systems can be used for the implantation of small implants. That's the idea that was suggested by the results obtained in the levothyroxine implant animal experiments. So a potential technology that can be used for this is microneedle technology. Okay, microneedle technology can be used to implant little or small implants made of biodegradable polymers like PLGA. Here you can see an example of a needle, and needles can be done with PLGA tips, and or they can even be loaded with long-acting suspensions. Okay, for doing this type of applications. Obviously, this is a topic for another day, because I'm not the right person to deliver this talk, because the person to deliver this talk is my colleague, Professor Ryan Donnelly, that works in the same school than I do. He's the expert on microneedle technology, but this is like a nice collaboration that we have ongoing, and he has like a track record on developing of these long-acting mini implantable systems. if you're interested in the details that I described before, not about the microneedle or anything like that, that's like, as I mentioned, that's like a topic for another day. But if you're interested in more specific details about biodegradable implants for like levothyroxine delivery, the results that I presented before, you can find more details in the following paper. It was published this year in International Journal of Pharmaceutics, and it was like a really nice collaboration with our colleagues in Indonesia. Okay. Additionally, there is like researchers from other institutions like Ulster University, and we got like this really nice publication. And in addition to this, we got more publications focused on the analytical methods used to quantify levothyroxine. Especially, we get quite pride in stabilizing the molecule for prolonged periods of time in aqueous solutions, and we got our publications out of that. These are the articles that were published about the analytical method. This one is focused on the in vivo analytical method of levothyroxine because it describes a simple method to quantify levothyroxine in plasma for preclinical studies without using complex to use or like approaches that require solid phase extractions or mass spec. It requires the use of a UV invisible detector in the chromatography. And this paper describes the method used to stabilize the drug and the HPLC method that was used to quantify. So if you work with levothyroxine and you are having stability issues, this paper, it's quite interesting because it describes a method that can be used to make your life easier in terms to stabilize the drug in solution. In addition to implants for drug delivery, for sustained drug delivery, we are interested in other type of medical devices, and we have done some research on them. Basically, one of the conditions that we are interested in addition to chronic conditions is cardiovascular disease. Okay, one specific application that we explore is the development of vascular grafts. Okay, vascular grafts are like devices or like systems that are used to bypass blocked arteries. Okay, they can be in the legs of a patient or they can be in the coronary arteries. Okay, whenever the blood vessel it's blocked or about to be blocked, surgeons can apply a bypass. Normally, this blood vessel that is used is taken from the harvest from the patient. Okay, and sometimes it's like a synthetic one. Okay, there's commercially available synthetic vascular grafts. But the use of a small diameter, smaller than 6 millimeters in lumen, vascular grafts can be problematic. Okay, because these new grafts tend to be blocked. These grafts, as I mentioned, they tend to be blocked, or they can be blocked because of the buildup, for example, of platelets they attach to the surface. This happens especially, as I mentioned before, in synthetic vascular grafts. So there is an interest on developing antithrombotic vascular grafts, synthetic ones, okay, because they can be adapted to the anatomy of the patient, and vascular grafts that can prevent the attachment of platelets to the surface of the material. For this purpose, we go back to the use of 3D printing because it's allow us to tweak the geometry of the implants and tweak the composition really easily. So for this purpose, we choose a biodegradable material because the vascular graft can be used to provide like a structure for cells, for endothelial cells or like cells from the cardiovascular cells to grow and regenerate the blood vessel. And additionally, we included anticoagulant drugs there to prevent the formation of blood clots in the meanwhile. So we choose two different drugs for this purpose, dipyridamole and aspirin. Okay, these two drugs were loaded and combined with this biodegradable material that was polycaprolactam. So whenever we started with aspirin, okay, aspirin was incorporated into the grafts, and we achieve drug loadings of up to 10%. We couldn't go higher because of the aspirin couldn't be used for printing because it blocks the nozzle. So 10% was the maximum concentration that was used for this purpose. Interestingly, when the drug was combined with polycaprolactone, we discovered that it was, as you can see in this DSC, and it was amorphous. It was well incorporated into the material. And you study the release profiles. You see that the difference between incorporating 5% or 10% in terms of cumulative release, it was similar. So 10% presented a slightly higher drug release, but obviously because of the different drug loading, the amount of aspirin released was higher whenever 10% was incorporated rather than 5%. This was translated, obviously, in a clear anticoagulant effect. Vascular grafts containing 10% of aspirin presented lower adherence of platelets to the surface whenever the materials were incubated with blood, and the platelets of the material were counted. So we discovered that materials or vascular grafts prepared containing 10% of aspirin, but they were resistant to the formation of, to the attachment of platelets. That subsequently this will lead to build up of blood clots. On the other hand, we work with dipyridamole. Dipyridamole is, like, in this case, has color, is yellow, it's a fluorescent molecule, and we achieve higher drug loadings. Okay, the drug was even up to 20%, it's the maximum concentration that was tested, 20% dipyridamole inside the implant. It was tested using computer micro CT. You can see that the drug was well dispersed in the material, as you can see in the image and in the CT images, so there was no accumulations of the drug or drug particles in any part of the vascular graft. Additionally, we can see that these materials were capable of providing a really sustained delivery of dipyridamole. We tested it for 30 days, but just the percentage of the total content in the graft was only, in the maximum loading was only 2% of the loading. So interestingly, there is not a massive difference in terms of the amount of dipyridamole release from these grafts, considering that the initial concentrations of drug in the graft were different. And what we observed is, like, the materials containing 20% dipyridamole showed a clear reduction in platelet adherence to the material. Okay, considering that the amount of dipyridamole release from these materials is almost the same, we can conclude that it's more important, in order to prevent the attachment of platelets in these materials, the amount of dipyridamole in the surface of the material is more important than the one that is released outside. Okay, so obviously we compare, like, materials made just with 3D printing and without 3D printing. We prepare some molds to compare, and we get virtually the same effect. So we can conclude that the 3D printing is not affecting the properties of the material. And actually, as you saw in those images, 3D printing can be used to prepare, like, vascular grafts containing up to 20% of drug loading in the case of dipyridamole. We tested, in this case, dipyridamole presented more interesting properties and better mechanical properties than the vascular graft prepared using aspirin. So we tested the mechanical properties of the resulting grafts and the hemocompatibility of the materials, showing that they were highly hemocompatible. And obviously they presented, because of the nature of PCL, polycaprolactone, that was the polymer used to prepare those grafts, we can see that the incorporation of dipyridamole reduced the ultimate tensile strength and the strain as failure. But even in that case, the resulting materials were acceptable and actually showed superior properties to actually native blood vessels. And actually, they showed superior mechanical properties than some of the material that has been described in the literature to prepare vascular grafts. Finally, we tested the compatibility with cells, with cells in the surface of the materials, and we can see that the cells proliferate, and there was not a big difference in terms of, like, the cell growth whenever we compare, like, with blank grafts. So the presence of the drug is not going to prevent the the proliferation of cells in the surface of the material. So that shows that these materials can be promising as scaffolds for regenerate vascular tissue. The work described about the development of 3D printed vascular grafts was published in included in two different publications. One in the journal Pharmaceuticals, that was focused on the development of aspirin-loaded vascular grafts, and another one published in Material Science and Engineering C. In this case, this work was focused on the development of dipyridamole-loaded vascular grafts. So if you're interested, you can have a look there. In this case, both publications are open access, so you should not have any problem. Just in case you want to have a look at the results. That's all what I have to talk for today. So thank you very much for your attention. If you have any question or you're interested in the work that we do and you want to collaborate, or you have any question, you have my email address there. Additionally, you can follow me on Twitter if you want to be updated, to be up to date with the things that on the research and the publications and the things that we are doing in the group. And thank you very much for your attention and for your time. Obviously, I cannot have done this without my awesome team, that the one there just supporting all the work and doing all the hard work and providing ideas and having a really nice work environment. And obviously this could not have been done without the support of Professor Donnelly, his microneedle team, and actually other collaborators that are not in the team, for example my colleague Dr. Andidian Permana from Indonesia, that is helping us massively in some of his research experiments. And obviously this could not have been possible without the support of some funding bodies that are providing some funding for the work that we do in the group.